Welcome back to the deep dive. I want to start today by asking you to do something a little weird.
Okay, I'm intrigued.
I want you to just look at your hand. Seriously, hold it up and look at it.
All right,
To you, it looks like skin, maybe some fingerprints, a hangnail. It looks like you.
Yeah,
A solid, single thing.
but it's not, is it? If we could zoom in, really zoom in with a microscopic lens, that hand would look like a planet.
A planet, exactly. It would be teeming with life.
Absolutely. Alien life from our perspective.
Yeah, I was reading the intro to Murray's chapter 7, and he has this whole concept of the microbial world, and it's basically that. We aren't just, single organisms walking around.
The ecosystems. Walking, talking ecosystems.
Covered in bacteria, fungi, viruses. They're in our gut, all over our skin, in our mouths.
It's a truly staggering thought, isn't it?
Wow.
That we are the host for literally trillions of other organisms.
Trillions.
And Murray makes this critical point right at the beginning.
Yeah.
Most of them, the vast, vast majority, are actually good guys.
The normal flora.
The normal flora. They're like tenants who pay rent. They take up real estate, so the actual bad guys, the pathogens, can't find a place to move in.
Right. But that's what gets me. If we're just covered in germs, and the world is full of them, how are we not sick all the time? I mean, I got a paper cut this morning. Technically, that's a breach of the fortress wall, right? So why am I not septic right now?
Because of the sanitation department?
I loved that line in the book. It's such a great way to put it. We always think of the immune system as, like, the military.
The defense forces, yeah. And it can be, for sure.
Yeah.
But 99% of what it does, day in and day out, is just sanitation.
Kicking out the trash.
Exactly. Cleaning up dead cells, sweeping away little bits of debris, and just, making sure the normal floras stay in their designated neighborhoods. It's maintenance.
So disease is when the sanitation workers go on strike.
That's one way. The trash piles up, pathogens move in, or the sanitation workers get way too aggressive and start throwing trash cans through the windows of healthy businesses.
And that's autoimmunity.
That's autoimmunity. It's all about balance.
So today, we are going to pop the hood on this whole system. We are diving deep into Chapter 7 of Murray, Elements of Host Protective Responses. We're going to map out the whole battlefield.
Meet the soldiers, learn the language they speak.
And we're going to pull in some key concepts from Jowett's Chapter 8 to give it that clinical spin because, this isn't just an academic exercise.
Absolutely not. We're learning this because when this system breaks, people get very, very sick. We need to understand the why behind it so we can figure out how to fix it.
Okay, so let's start with the big picture. In med school lectures, you always hear about the immune system as if it's one thing, but it's really two, right?
It's two completely different armies, the innate and the adaptive. That's the first and most fundamental split you have to understand.
So innate is the old one.
Ancient. Think starfish and insects. It's the system that's been around forever, and its characteristics are really important. It's rapid, it's totally nonspecific, and it has zero memory.
So it's like a security guard with a flashlight in a dark warehouse.
That's a perfect analogy. It just sees something moving in the dark and it whacks it.
Doesn't care if it's a burglar or a raccoon.
Nope. Same response every single time. And if that same burglar comes back a week later, the innate system has no idea it's seen him before. It's just intruder alert all over again.
And the adaptive system, that's the new fancy one.
That's the sniper team. It's highly, highly specific. It takes days, sometimes weeks to get going because it's studying the target. It learns the specific face of that one enemy.
And the crucial part is the memory.
The memory is everything. If that specific enemy ever dares to show its face again, the adaptive system takes it out before you even feel a sniffle. That's the entire principle behind vaccines.
Okay, so we have the brute force guards and the elite assassins. But before any of them even show up, something has to sound the alarm. Murray has this phrase I liked. Soluble before cellular.
Yes. This is a concept that students often trip over. We get so excited about the cells, the cool macrophages and neutrophils, that we forget the first response is chemical.
The environment has to change before the troops arrive.
Precisely. The chemical signals go out first.
So walk me through it. Let's go back to my paper cut, splinter, whatever. The barrier is broken. What is the very, very first thing that happens?
Okay, so even before that paper sliced your skin, you had chemical defenses already in place. Your skin isn't just a passive barrier like plastic wrap.
It's chemically active.
Exactly. You have an enzyme called lysozyme in your sweat, in your tears. Its whole job is to dissolve bacterial cell walls. Then you have these amazing little peptides called defensins.
Defensins, what are those?
Think of them like tiny molecular spears. They're positively charged, And bacterial membranes are negatively charged, so they're drawn to them.
Opposites attract.
Right. And they just insert themselves into the membrane and poke holes in it. It's like walking through a minefield for any bacteria trying to set up shop on your skin.
Okay, but my paper cut got past the minefield. The bacteria are in the deeper tissue now, so what happens?
Now the tissue screams.
It screams.
Chemically. When your own cells get damaged, they burst open and spill their guts. Or when bacteria start setting up camp and metabolizing, they release molecules. All of this chemical noise brings us to the most important communication network in the body.
Cytokines and chemokines.
The language of immunity.
I feel like these two words get used almost interchangeably. You hear about a cytokine storm in severe infections, but they are different.
They are very different. And for any exam, and just for, logically understanding how this all works, you have to keep them separate.
Okay, break it down for me.
Think of a cytokine as a command. It's a protein, kind of like a hormone, that tells a cell what to do.
Like wake up, or start dividing.
Or start making poison, or commit suicide. It's an order.
And a chemokine.
A chemokine is a map coordinate. It's a chemoattractant. It tells the cell where to go.
Okay. So the cytokine is the radio dispatch saying, we have a code red, all units respond.
Right. And the chemokine is the GPS signal pinging the exact location of the fire.
I get it. The cells literally follow the signal.
They do. Chemokines create a concentration gradient, like a breadcrumb trail. The immune cells smell their way from low concentration to high concentration, right to the source of the infection.
Murray gives us a great mnemonic for thinking about any cytokine stat, which I'm guessing is not just because doctors like to yell that in the ER.
No, but it is easy to remember that way. So, S is for source. You always have to ask, who sent this message? Was it a macrophage, a T cell, a skin cell?
Okay, source.
T is for trigger. Why was this message sent? Did the source cell see a piece of a virus or a bacterial toxin? What was the stimulus?
Makes sense.
A is for action. What is the actual order? Does it cause fever? Does it cause inflammation? Does it tell another cell to grow?
And the last T?
T is for target. Who is supposed to be listening to this message?
That seems really important. You don't want everyone in the body getting every single message.
You absolutely don't. If you release a cytokine that says commit suicide, apoptosis, you only want the virus-infected cell to get that message, not the healthy liver cell right next to it.
So how does that work?
The specificity of the receptor on the target cell, it's like a locking key.
Yeah.
Only the cells with the right receptor can hear that specific cytokine. That's the safety mechanism.
Okay, that makes sense. There's another acronym Murray gives us for the bigger picture, for the whole story of an immune response. Tai-2-Talk.
Right. Tai-2-Talk is a framework to help you build the narrative of any infection from start to finish.
Because time is of the essence.
Exactly. So T is for trigger, the initial event, the paper cut, the inhaled virus. I is for inducer. This one is super important. The immune system doesn't see a salmonella bacterium the way our eyes do.
What does it see?
It sees molecular patterns. We call them PAMPALs, pathogen-associated molecular patterns.
KMATs, like what?
Like LPS, lipopolysaccharide. It's a molecule on the outer surface of all gram-negative bacteria. Your immune cells have receptors that are hardwired to recognize LPS as danger.
So it's like seeing the enemy's uniform, not their face.
Perfect analogy. Or it can see damage-associated molecular patterns. That's bits of your own cellular guts spilling out when a cell dies a messy death. The system sees that and knows something is wrong.
Okay, so trigger, inducer. C is for cells.
Yep. Which cells are producing the signals and which cells are responding to them.
Then T for time course.
Is this an acute response happening over minutes to hours, or is this a chronic thing that's been smoldering for months?
O is for outcome.
Did we win?
Yeah.
Is the infection resolved and the tissue repaired? Or did we lose and now we have pathology and disease?
And the final C
is for cytokines. Which specific chemical mediators are driving this whole process? If you can answer all those Tai-2-Talk questions, you understand the infection.
I really like that. It turns it from a list of facts to memorize into a story.
That's the whole point.
Okay, but we have to talk about the real heavy artillery of the soluble world, the complement system.
Ah, yes, the bane of every first-year medical student's existence.
It really is. C1, C3A, C5B, C9. It feels like a game of bingo where you never, ever win.
It seems incredibly complicated, and the details can be, but the core concept is actually very elegant.
Please make it elegant for me.
Think of the complement system as that pre-placed minefield we talked about. It's a set of over 30 proteins floating around in your blood, completely inactive all the time.
Just waiting.
Just waiting. But if one of them gets triggered, it activates and cleaves the next one, which activates and cleaves the next one. It's a cascade, an amplifying enzymatic cascade.
And there are 3 different ways to trip that first wire, right?
3 main pathways, yes. There's the classical pathway, which is triggered by antibodies. So this is the adaptive system calling in an airstrike from the innate system.
Okay, antibodies start that one.
Then there's the alternative pathway. This one is triggered spontaneously just by the surface properties of pathogens themselves. It's like a motion sensor. It's always sort of ticking over at a low level, and when it bumps into a bacterial surface, it goes off.
So that's pure innate.
Pure innate. And the third is the lectin pathway, which is triggered by binding to specific sugars like mannose that are common on microbial surfaces but not on our own cells.
Okay, so classical, alternative, lectin. That's how it starts.
But here's the secret. For your exams and for your sanity, don't obsess over the start of the pathways.
Focus on the end.
Focus on where they all meet. They all converge on one critical step. The splitting of a molecule called C3.
C3.
Once you split C3 into C3A and C3B, the whole game changes. This is probably the highest yield part of the whole system. You have to know what C3A, C3B, and C5A do.
Okay, let's break them down. What about the A fragments, C3A and C5A?
The A stands for anaphylatoxin. Think of them as the sirens and the fire alarms. They are potent inflammatory mediators.
What do they do?
They cause vasodilation. They make your blood vessels widen and, more importantly, become leaky.
Why on earth would you want leaky blood vessels?
Because your best soldiers, the neutrophils, are inside the blood vessels. To get to the fight in the tissues, they need to be able to slip out between the cells of the vessel wall. C3A and C5A open the doors for them.
Oh, okay, so they're recruiting help.
Massively. And C5A is also one of the most powerful chemokines known. It's that breadcrumb trail we talked about, pulling neutrophils directly to the site of invasion.
So C3A and C5A are the alarms. They scream for help and open the doors. What about C3B?
C3B is totally different. It's not a signal, it's a weapon, or maybe a tool. It binds directly, covalently to the surface of the pathogen.
It tags the bacteria.
It tags it. We call this process opsonization.
That's a Greek word, right?
It is. It means to prepare for eating.
Ha! I love that.
And it's so descriptive. Here's the thing. Bacteria are often slippery. Many of them have these slimy capsules. A phagocyte, like a macrophage, tries to grab on, and the bacterium just squirts away.
Like trying to catch a greased pig.
Exactly like that. C3B acts like a handle. It coats the bacteria in these little tags that the phagocytes have receptors for. It's like putting Velcro on the pig. Now the macrophage can get a firm grip and eat it.
So opsonization is basically making things tastier for the immune cells.
Or at least easier to catch. It's a critical step. Without opsonins like C3B and antibodies, phagocytosis is really inefficient.
Okay, but what if that's not enough? What if the bacteria are too tough? Does complement have a nuclear option?
It does, the MAC.
The membrane attack complex.
This is the end of the line. C5b, which is also generated, kicks off a new assembly line. It recruits C6, C7, C8, and then a whole ring of C9 molecules.
What do?
They literally punch a hole. They form a tube, a pore, that stabs directly through the bacterial cell membrane.
Like drilling a hole in a submarine.
That's the perfect image. Water and salts rush in, the internal environment is destroyed, and the bacterium just leases. It explodes.
Wow. That is violent.
It is. And what's amazing is that this all happens automatically. No cells had to give an order. It's pre-programmed chemistry. That's the power of the soluble innate system.
All right, so the chemical alarms are ringing, the mines are exploding. It's time to call in the troops.
The cellular response.
Let's move to section 2, the cast of characters. Murray gives us another framework here called CRRP to help us keep all these cells straight. Because honestly, looking at them on a slide, they all just kind of look like little purple blobs.
They really do. But it's their function that matters. So CRP stands for C is for cell surface markers. We call them CD markers.
Cluster of differentiation.
Right. A is for actions. What does the cell actually do? Does it eat things? Does it kill things? Does it send signals?
R is for role. Is it part of that ancient innate army or the fancy adaptive army?
And finally, P is for products. What does it secrete? Does it make cytokines? Does it make antibodies?
The CD markers, they're basically like the uniforms or ID badges for the cells, right?
That's exactly what they are. Doesn't matter what a cell looks like under a microscope. If you can stain it and see that it's wearing a CD4 badge, 100%, that it's a helper T cell.
And if it has CD19?
It's a B cell. Memorizing the key CD markers is just non-negotiable because that's how we identify and count these cells in the lab in real patients.
Got it. Okay, before we meet the individual soldiers, we need to visit the factory and the training grounds. Section 3, hematopoiesis And the anatomy of defense.
The origin story.
It all starts in the bone marrow.
Everything. All of your blood cells, red cells, white cells, platelets, they all arise from a single common ancestor, the pluripotent hematopoietic stem cell.
The great-grandmother of the entire immune system.
She is, and she has two main daughters. This is the biggest fork in the road in all of immunology.
The myeloid progenitor and the lymphoid progenitor.
That's the split. The myeloid lineage gives rise to what you can think of as the blue-collar workforce.
Like who?
Your red blood cells, your platelets, and the real grunts of the immune system. The neutrophils, monocytes, eosinophils, and basophils. They're produced in staggering numbers. We make billions of neutrophils every single day.
Billions. And the lymphoid lineage.
That's the white-collar crowd, the specialists, the special forces, your T cells, your B cells, and your natural killer, or NK cells. They're generally fewer in number, they live much longer, and they are highly, highly educated.
Okay, speaking of education, Murray makes a big distinction between primary and secondary lymphoid organs. This seems important.
It's critically important. Think of the primary organs as the university or boot camp. This is where the cells are born and where they get their education.
There are only two of them.
Only two, the bone marrow and the thymus.
Okay.
In the bone marrow, B cells are born and complete their maturation. It's also, of course, the source of all the stem cells. In the thymus, T cells go to get their education.
And what's on the curriculum at Thymus University?
One subject, and it's pass-fail. Self versus non-self.
How do they teach that?
It's a brutal process.
Yeah.
In the thymus, these young T cells, called thymocytes, are shown little pieces of your own body. They're shown bits of your own liver proteins, your heart proteins, your skin proteins.
A test.
A loyalty test. And if a T cell recognizes and attacks one of those self-proteins, the thymus kills it. It forces it to undergo apoptosis.
Wow.
It's an intense safety check. The message is, if you attack the host, you die. Something like 95% of all T cells that enter the thymus fail this test.
95% flunk out.
They do. They're destroyed. Only the tiny fraction that prove they are safe, that they won't attack you, are allowed to graduate and enter the circulation.
And that's to prevent autoimmune disease.
Absolutely. If even one of those autoreactive cells escapes, it can wreak havoc.
Okay, so they graduate from university. Where do they go to work?
They go to the secondary organs.
Yeah.
These are the battlefields with the police stations. This is where the mature, naive lymphocytes hang out, waiting for a call to action.
And these are the lymph nodes.
The lymph nodes, the spleen, and something called malt mucosa-associated lymphoid tissue. Think tonsils, Peyer's patches in your gut.
And the whole point of these places is to bring everyone together.
Exactly. Their entire structure is designed to maximize the probability that an antigen, a dendritic cell carrying that antigen, and the 1 in a million lymphocyte that can recognize that antigen will all meet in the same place at the same time.
It's a matchmaking service.
An immunological matchmaking service. Precisely.
Okay, let's zoom in on the lymph node. Murray calls it a sewage processing plant, which is a vivid image.
It's gross, but it's accurate. Think about it. Fluid is constantly leaking out of your capillaries into your tissues. That fluid is called lymph. It has to be collected and returned to the blood.
So it gets sucked up into lymphatic vessels.
Right. And on its way back to the heart, it gets filtered through these lymph nodes. So if you get a splinter in your foot, any bacteria that get in get swept up in the lymph and are delivered directly to the lymph node in the back of your knee.
The lymph node is a trap.
It's a beautiful, highly organized trap. And you need to be able to visualize its architecture. Think of it like a kidney bean or a target with distinct rings.
Okay, what's the outer ring?
The outer ring is the cortex. This is B-cell territory. You'll see these round structures here called follicles. When B cells get activated by an infection, these follicles swell up and develop hot zones of proliferation in the middle. We call those germinal centers.
Germinal centers. That's where they're basically cloning themselves to fight the infection.
Yes. They're dividing like crazy, and they're also intentionally mutating their antibody genes to try and make an even better, stickier antibody. It's called somatic hypermutation.
Wow. Okay, so cortex is for B cells. What's the next layer in?
The middle ring is the paracortex. This is T-cell territory. This right here is arguably the most important room in the entire immune system.
Why?
This is where the dendritic cells, that scouts will talk about, come running in from the tissues to present what they've found to the T-cells. The whole adaptive response starts here, in the paracortex.
And the very center of the node.
That's the medulla. You can think of this as the exit lobby.
Okay.
This is where you find a lot of macrophages cleaning things up. And crucially, Plasma cells.
Plasma cells are the activated B cells, the antibody factories.
Exactly. They've been activated, they've turned into factories, and now they've moved to the medulla to just pump thousands and thousands of antibodies into the efferent lymph, which then dumps into the blood.
So let me see if I have the flow right. An antigen comes in through the afferent lymphatic.
Yep.
A dendritic cell catches it, runs to the paracortex to wake up a T cell.
Perfect.
That T cell then helps wake up a B cell over in a follicle in the cortex.
Right.
The B cell turns into a plasma cell, moves to the medulla, and starts shooting out antibodies that leave the node.
You've got it. That's the assembly line of adaptive immunity right there.
What about the spleen? Is that just a giant lymph node?
It's similar, but with one massive difference. A lymph node filters lymph fluid coming from the tissues. The spleen filters blood. This makes it absolutely vital for dealing with bacteria that get directly into your bloodstream bacteremia, especially bacteria that have slimy capsules that help them evade the immune system.
So like meningitis or certain pneumonias.
Exactly. Neisseria meningitis, streptococcus pneumonia. The spleen is where they get caught. It has areas of white pulp, which is the immune tissue, and red pulp, which is where old, damaged red blood cells are taken out of circulation.
And in the white pulp, is the geography similar?
It is. Around the central arterioles, there's a cuff of T cells called the periarteriolar lymphoid sheath. That's the spleen's version of the paracortex. And then you have B-cell follicles surrounding that.
So a good memory aid is that there's no T in follicle. That's a B-cell zone. But there are Ts in paracortex and in the periarteriolar lymphoid sheaths.
That's a great one. I like that.
So clinically, what happens if someone gets in a car crash and they have to have their spleen removed? A splenectomy.
They lose their primary blood filter. They become incredibly susceptible to overwhelming sepsis from those encapsulated bacteria we just mentioned.
So they need special care.
Absolutely. They need an extra slate of vaccines against those specific bugs, and often they're put on lifelong prophylactic antibiotics because they've lost a key piece of their defensive anatomy.
It really drives home that these organs aren't just redundant parts. Okay, let's finally meet the troops. Section 4, the myeloid lineage, the boots on the ground.
The first responders.
First up, the most famous one, the neutrophil, also called a PMN.
Polymorphonuclear leukocyte. The infantry. These cells are the most abundant white blood cell in your circulation, maybe 60 to 70%.
And they are built for one thing.
Kill and die. They have an incredibly short lifespan, maybe a day or so. They are programmed to get to the site of infection as fast as possible, release all their toxic granules, eat a few bacteria, and then die.
And a pile of dead neutrophils is?
That's what pus is. It's mostly dead neutrophils, dead bacteria, and liquefied tissue.
Lovely. But they have this really cool diagnostic feature that doctors are always looking for on a blood test. The left shift. What is that?
This goes right back to the bone marrow factory. A fully mature, ready-for-battle neutrophil has a nucleus that's segmented into 3 or 4 distinct lobes. We call those segs.
Okay, segs are the grown-ups.
Right. An immature neutrophil, like a teenager still in training, has a nucleus that hasn't segmented yet. It just looks like a curved band or a sausage. We call those bands.
Bands are the recruits.
Exactly. Normally, the bone marrow keeps the bands in reserve and only lets the mature segs out into the blood. But if you get a massive bacterial infection, the demand for neutrophils skyrockets. The marrow panic.
It needs bodies on the front line.
It needs warm bodies now. So it starts pushing the teenage bands out into the circulation before they're fully trained. So if we look at your blood smear and see a high percentage of these immature bands, we say the population has shifted to the left towards immaturity on the developmental chart.
And that's a huge clue.
It's a classic sign of an acute bacterial infection. It's mechanism-based reasoning at its best.
Neutrophils also have this totally metal way of dying, right? Nets.
Ah, yes. Neutrophil extracellular traps. This is one of the coolest things in immunology.
It sounds cool.
So imagine a neutrophil gets to the battle. It's surrounded. It knows it's going to die. As a final suicidal act, it decondenses its own chromatin, its own DNA.
It dissolves its nucleus.
Basically, yeah. And then it explodes, spewing out this web of sticky DNA.
A net made of DNA.
A net made of DNA. And this web physically traps bacteria, preventing them from spreading. And to make it even better, the web is decorated with all of the neutrophil's toxic granule proteins. So it's a sticky, poisonous net.
That is unbelievably cool. The cell sacrifices itself to build a barricade.
It's incredible.
Okay, what about the other granulocytes, eosinophils and bellophils?
These are more specialized. The neutrophils are the infantry. These are the specialists for things you can't eat. like a giant parasitic worm.
You can't phagocytose a worm.
You can't.
Yeah.
So an eosinophil comes along, it has a bilobe nucleus and stains bright red with eosin dye, and it just snuggles up to the worm and releases its toxic granules all over the worm's surface, trying to poison it from the outside.
And basophils.
Basophils and their tissue-resident cousins, mast cells, are the chemical warfare units. Their granules are packed with things like histamine. They're the major players in allergic reactions, type hypersensitivity.
Okay, now for the big eaters. The heavy hitters. Monocytes and macrophages.
The Pac-Man of the immune system. The monocyte is the form that circulates in the blood. It's like the cell is commuting.
And when it gets to its stop?
When it gets the chemokine signal, it exits the blood vessel, crawls into the tissue, and undergoes a transformation. It swells up, gets way more aggressive, and becomes a macrophage, a big eater.
And unlike the neutrophils, these guys stick around.
For a long time. Macrophages can live for months or even years in tissues. They're not just soldiers. They're resident peacekeepers and construction workers.
And they can change their personality, right? This M1 versus M2 switch seems really high yield.
It's a huge concept. A macrophage isn't just a macrophage. It can be polarized into one of two main states.
M1 is war mode.
M1 is war mode. It's activated by cytokines like interferon gamma. An M1 macrophage is angry. It's producing reactive oxygen species, basically bleach and nitric oxide.
Yeah.
It's highly inflammatory and fantastic at killing bacteria.
And M2?
M2 is peace mode, tissue repair mode. Once the battle is won, the macrophage can switch its phenotype. It starts secreting growth factors that promote wound healing, new blood vessel formation, and cleaning up debris.
What happens if that switch breaks, if you're stuck in M1 war mode forever?
That's the basis of many chronic inflammatory diseases. Think about rheumatoid arthritis, where macrophages are just endlessly attacking the joint lining, or Crohn's disease in the gut. You're constantly bombing your own city instead of rebuilding it.
And just to make our lives harder, macrophages get different names depending on which tissue they live in.
They do. In the liver, they're called Kupffer cells.
In the brain.
Microglia.
In the bone.
Osteoclasts. But they're all part of the same family, the mononuclear phagocyte system.
One last thing on macrophages. How do they know what to eat? We talked about opsonization, but do they have their own sensors?
They do. They have a whole panel of receptors called pattern recognition receptors that recognize those PMPs we mentioned earlier. The most famous one is CD14, which is part of the receptor complex for LPS from gram-negative bacteria.
So they have a nose for bacteria.
A very good one. And they have FC receptors, which are the hands that grab onto the antibody handles that are coding a pathogen so they can see things directly or see them once they've been tagged by the adaptive system.
Okay, this brings us to a really key player. Section 5, the director. The dendritic cell. Why do we need another phagocyte if we already have these amazing macrophages?
Because they have totally different jobs. The macrophage is a beat cop. Its job is to handle local crime, clean up the mess at the site of infection.
And the dendritic cell.
The dendritic cell, or DC, is an intelligence officer. Its primary job is not to clear the infection. Its job is to document the infection, gather intelligence, and carry that intel back to headquarters.
Headquarters being the lymph node.
Exactly. And here is the golden rule you have to burn into your brain. A naive T cell can only be activated by a dendritic cell.
Not by a macrophage.
No. A naive T cell is like a 4-star general. It's too important to be bothered by a beat cop. It demands a formal briefing from a dedicated intelligence officer. The dendritic cell is the only cell that can provide that briefing and kickstart a primary adaptive immune response.
So the DC eats the bacteria in the skin.
Chews it up, processes it, and puts a little piece of the bacterium on its surface, displayed on a molecule called MHC, and then it leaves.
It abandons the battlefield.
Completely. It migrates out of the tissue, finds a lymphatic vessel, and travels to the draining lymph node. It walks right into the paracortex and starts looking for the one T cell that can recognize the intel it's carrying.
That bridge from the innate world to the adaptive world is the DC.
It is the indispensable bridge. Without it, the adaptive response never starts.
Okay. So let's cross that bridge. Section 6. The lymphoid lineage. The special forces. Let's talk T cells.
The managers and the assassins of the adaptive immune system.
They all wear the CD3 badge, right? That's the universal T cell marker.
Correct. CD3 is part of their antigen receptor complex. If it's CD3 positive, it's a T cell. But then they split into two major divisions based on a second badge.
CD4 and CD8.
Right, and they have fundamentally different jobs.
CD4 cells are the helper T cells.
I like to call them the generals or the dispatchers. They don't carry weapons themselves, they carry radios. Their job is to assess the intelligence from the dendritic cell and then coordinate the correct type of battle plan.
So the DC shows them a piece of the enemy, and the CD4 cell decides what to do about it.
Precisely. If the intel says we're dealing with an intracellular bacteria or a virus, the CD4 cell will differentiate into a Th1 cell. It then sends out cytokines that scream for macrophages and CD8 killer cells, cell-mediated immunity.
Okay, Th1 for viruses. What if it's a parasite?
If the intel says we have a big worm, the CD4 cell becomes a Th2 cell. It sends out a completely different set of cytokines that scream for B cells to make antibodies and for eosinophils to show up. Humoral immunity.
So there are different flavors of helper T cells for different kinds of threats.
Yes. There's also Th17 for fungal infections, which calls in neutrophils, and there are TREG, or regulatory T cells, whose job is to suppress the immune response and turn it off when the war is over. The CD4 cell is the strategic mastermind.
And getting that strategy wrong is a disaster.
It's the difference between health and disease. In leprosy, for example, if your body mounts a Th1 response, you contain the bacteria, get a mild form of the disease. If your body mistakenly mounts a Th2 antibody response, it's completely ineffective, and the bacteria grow uncontrollably. The general chose the wrong strategy.
Wow. Okay, so what about the CD8 cells?
These are the cytotoxic T lymphocytes, the assassins, the cops on the beat.
What's their job?
Their job is to patrol your body and check the ID cards of all your own cells.
The ID card is the MHC molecule.
Specifically, MHC class I. Every single nucleated cell in your body has MHC class I on its surface, and its job is to act like a little display window, showing a sample of all the proteins currently being made inside that cell.
It's like a quality control check.
A constant one. If a cell is healthy, it just shows little bits of normal human proteins. The CD8 cell glances at it and moves on. But if that cell gets infected with a virus, it will start making viral proteins.
And a piece of that viral protein gets displayed in the window.
Exactly. The CD8 T cell comes along, sees the foreign protein in the MHC class I window, and knows that cell has been compromised. It locks on and delivers the kiss of death.
Which is what, exactly?
It's a payload of two key proteins, perforin and granzymes.
Perforin word, perforate.
It punches a hole in the target cell's membrane. Granzymes are enzymes that then enter through that hole and flip the switch for apoptosis. They tell the cell to commit suicide.
And it does so neatly so the virus can't spread.
It just implodes, and the mess is cleaned up by macrophages. It's an incredibly elegant and precise way to eliminate infected cells without causing a lot of collateral damage.
Okay, last of the main lymphocytes. B cells. The factories.
B cells are all about humoral immunity. Immunity mediated by antibodies in the humors or fluids of the body.
So when they get activated, usually with help from a CD4 T cell?
They undergo this incredible transformation. The naive B cell becomes a plasma cell.
And if you look at a plasma cell under a microscope...
It's amazing. The nucleus is tiny and pushed off to one side. Why? Because the entire cytoplasm is just jam-packed with rough endoplasmic reticulum.
Which is the cellular machinery for making proteins.
And in this case, that protein is antibody. A single plasma cell can secrete something like 2,000 antibody molecules per second.
2,000 per second.
Per second. It is a biological machine gun, just flooding your system with antibodies designed to find their target. They opsonize pathogens, they neutralize toxins, they activate complement. They are the product of the humoral response.
And just to close the loop on the lymphoid lineage, there are the NK cells, natural killers.
The NK cell is the backup plan, the evolutionary counter move.
To what?
Some viruses are smart. They know that CD8 T cells are looking for viral peptides on MHC class I. So the virus evolves a mechanism to tell the infected cell, hide your MHC, pull it inside so the T cell can't see you.
So the infected cell goes dark. It becomes invisible to the CD8 T cells.
To the T cells, yes. But the NK cell operates on the opposite logic. The NK cell patrols the body demanding to see an ID. It looks for the presence of normal MHC class I.
So if a cell doesn't show an ID...
If a cell has no MHC class I on its surface, the NK cell assumes it must be hiding something and it kills it. No questions asked.
So let me get this straight. The CD8 T cell kills if it sees a bad ID.
Correct. A foreign peptide in MHC.
And the NK cell kills if it sees no ID.
The absence of MHC, it's called the missing self hypothesis. Between the two of them, there's almost nowhere for a virus to hide. It's a brilliant system.
That is beautiful. It's like a perfectly designed trap. Okay, let's put it all together. Section 7. Synthesis, let's run a simulation. I step on a rusty nail. A bunch of Staphylococcus aureus gets into the wound. Go.
Okay. Time 0. Nail breaches the physical barrier, the skin.
First few seconds.
Bacteria are in the tissue. They immediately encounter complement proteins. The alternative pathway kicks on. C3b starts coating the bacteria. C3a and C5a are released into the tissue.
First few minutes.
Resident mast cells in the tissue sense the damage and the C3a. They degranulate, releasing histamine. Blood vessels in the area dilate and get leaky. The area gets red and warm.
First 2 hours.
Neutrophils cruising through those blood vessels smell the C5a and other chemokines. They slow down, stick to the vessel wall, and then squeeze through the gaps, a process called diapedesis.
They rush to the nail.
They follow the chemokine trail right to the bacteria. They start gobbling up the C3b-coated staph. They release their granules. Many of them die, forming that early pus.
Okay, so that's the innate response, happening fast. What about the adaptive side?
At the same time, a dendritic cell in that tissue has also been eating some of the staph bacteria. It gets activated, packs its bags, and starts its journey to the draining lymph node.
And this takes a few days.
Right. Maybe on day 3 to 5, that DC arrives in the paracortex of the lymph node. It finds the one specific naive CD4 T cell that happens to recognize that piece of staph. It activates it.
The general has been briefed.
The general is awake. That CD4 T cell then activates a specific B cell in a nearby follicle. That B cell starts proliferating, forming a germinal center.
Day 7 to 10.
Those B cells have now become plasma cells. They're pumping out huge amounts of high-affinity IgG antibody, specifically against Staph aureus. That antibody floods the bloodstream.
And the result?
The antibody travels all the way back to the toe, coats any remaining bacteria, neutralizes any toxins they're making, and the infection is definitively cleared. Then M2 macrophages move in to start the repair process.
It really is a symphony. Every part has its role.
It is.
But before we wrap, we have to touch on when the symphony is out of tune. Joetz has a good section on this. Hypersensitivity. This is when the immune system works too well or on the wrong thing.
Right. It's the dark side of this powerful system. You can break them into 4 types.
Type I is allergy.
Your mast cells and IgE antibody overreacting to something harmless like peanut protein or pollen.
Type 2.
Antibody against your own cells. You make antibodies that stick to your own red blood cells, for example, and then complement comes along and blows them up. Hemolytic anemia.
Type 3 is collateral damage.
Exactly. You get huge complexes of antigen and antibody that get stuck in small blood vessels, like in the kidney, and cause inflammation. That's what happens in lupus or post-strep glomerulonephritis.
And finally, type 4.
That's T-cell mediated, no antibodies involved. This is when your T cells get overexcited and cause damage. The rash you get from poison ivy or the bump from a TB test, that's a type 5E reaction.
It all comes back to balance. The sanitation department has to clean the street, not burn down the whole neighborhood.
That's the constant struggle.
Well, my brain is officially full. But we're not quite done. It is time to see if you were paying attention.
Time for the recall.
I'm going to ask a few rapid-fire questions, typical single best answer style. We'll have a brief pause, and then you give us the answer and, more importantly, the why.
Let's do it.
Question 1. You're looking at a patient's complete blood count with differential, and you notice a significant increase in neutrophils that have an unsegmented band-like nucleus. What is this finding called, and what is the fundamental process in the bone marrow that causes it?
Okay. That finding is called a left shift.
Yeah.
And the why is that a severe acute bacterial infection is causing such a high demand for neutrophils that the bone marrow is forced to release immature forms, the bands, from its reserves into the circulation prematurely.
Perfect. Question 2. A dendritic cell has just phagocytosed a pathogen in the skin and is migrating to the lymph node to activate a T cell. To which specific anatomical zone within the lymph node must this dendritic cell go to find and present antigen to naive T cells?
That would be the paracortex. Remember the geography. The cortex is for B cells. The nulla is the exit. The critical first meeting between the APC and the naive T cell happens in the T cell-rich paracortex.
Excellent. Question 3. We know that both cytotoxic CD8 plus T cells and natural killer, or NK cells, are capable of killing virus-infected host cells. What is the critical difference in their target recognition mechanisms?
Right, they both kill, but they're looking for different things. CD8 plus T cells recognize a specific viral peptide presented on an MHE class I molecule. They see a bad ID. NK cells, on the other hand, recognize the absence of MHE class I molecules on a cell surface. They kill cells with no ID.
Great distinction. Question 4. In the complement cascade, several fragments are generated with different functions. Which specific complement fragment is the primary opsonin, the one responsible for coating a pathogen to make it tastier for phagocytes?
The main opsonin is C3b, While C3A and C5A are the anaphylatoxins that cause inflammation and attract cells, C3b is the fragment that physically binds to the pathogen's surface and acts as the eat me signal for phagocytes.
All right, question 5. A patient who has had their spleen surgically removed, and a splenic patient, is at a significantly increased risk for life-threatening sepsis from what general category of pathogens and why?
They are most susceptible to encapsulated bacteria, bugs like strep pneumo, H. flu, and Neisseria meningitidis. The why is that the spleen is the primary site for filtering blood and clearing pathogens that are resistant to phagocytosis. Without the spleen's macrophage population, these slimy encapsulated bacteria can multiply in the blood unchecked.
Last one, question 6. A macrophage can exist in a resting state or be activated into a highly aggressive pro-inflammatory bacteria killing machine. What is the master cytokine that drives this polarization into the so-called M1 phenotype?
The master switch for the M1 phenotype is interferon gamma, IFN gamma. The cytokine, which is usually secreted by Th1 cells and NK cells, supercharges the macrophage's killing capacity, turning on its respiratory burst and making it an antimicrobial powerhouse.
Excellent. How did you do at home? If you found yourself struggling with some of the anatomy questions, I really recommend going back to Murray Chapter 7 and just staring at that diagram of the lymph node. It is high-yield real estate.
It really is. And remember, what we did today was just introduce the cast of characters and the map of the battlefield. In the next few deep dives, we're going to see how this army actually fights specific wars, how the strategy changes for a virus versus a fungus versus a parasite.
Until then, keep your barriers intact, wash your hands, and be nice to your sanitation department. Thanks for listening to The Deep Dive.
Stay curious.